Hello everyone, good evening. I'm John Calvin and welcome back to a brand new episode of BioTalks, powered by BTech Industries, co-powered by AB Builders. In this episode, we'll be talking to some of the famous scientists who led to the discovery of the cell. So, let me introduce Camilo Golgi, Christian J. Jouet, Rudolf Albert von Kolliker, George Emil Perret, Albert Claude, Anthony van Leeuwenhoek, Robert Brown, Robert Hooke, and Andreas Schimper to this stage. Firstly, I call upon Robert Brown to talk about one of the pioneers in the discovery of the nucleus. Welcome, Dr. Brown.
Hello, it's my pleasure to be here.
Sir, would you please tell us what led you to the discovery of the nucleus?
When you think about it, it actually took me a while. But while watching pollen grains fertilizing a plant, I noticed there were tiny ovals in the cells, and pollen was moving in and out of the ovals. I realized that they were of great importance to the cell, and I called them nucleus. So basically, what the nucleus does is, it is the main control center of the plant. It tells the cell what it should do. So this nucleus contains the genetic material. Genetic material is what defines the cell. So this material is embedded in the chromatin. Chromatin consists of DNA and proteins. So when I was watching this fertilization, I realized that this chromatin was arranged in specific patterns. So I decided to name them chromosomes. So chromosomes are basically the chromatin arranged in specific layers during fertilization. So basically that's it. That is the main idea of the nucleus.
Thank you, Dr. Brown, and that was very interesting indeed. Next, I would like to call upon Dr. Robert Hooke, who paved the way for modern biology.
Thank you, Mr. Calvin. It's an honor for me to be here.
Oh, sir, it's also a great pleasure for me to meet someone as great as you. Sir, could you please tell our audience about your contributions to biology?
Okay, so since you asked that, two of my main observations were of cell membrane and cell wall. Let me explain. When I was watering the plants, a thought came to my mind. Water components will be present in a big tree trunk. While observing cork through my microscope, I saw tiny box-like cavities, which I called cells. After many observations, I also found out the cell wall and cell membrane.
Thank you, Dr. Hooke, for spending some time with us. Now, I would like to welcome Rudolf von Kolliker to talk about the cytoplasm and mitochondria. Welcome, Mr. Rudolf.
Thank you, Mr. John. Two of my most important contributions was the discovery of cytoplasm and mitochondria. First of all, cytoplasm is all the material within a eukaryotic cell, enclosed by the cell membrane. Most cellular activities take place within the cytoplasm, such as many metabolic pathways, including glycolysis, and processes such as cell division. I discovered the mitochondria in 1857, and I referred it to as the powerhouse of the cell. This organelle is responsible for converting food molecules and oxygen into adenosine triphosphate, ATP. This ATP is used to give energy to the cell.
Thank you, Dr. Kolliker. It was a pleasure to meet you. Now, we shall go for a short break. This is BioTalks powered by BTech Industries, co-powered by AB Builders.
छक्का किस बात का आउट है भाई? छत के ऊपर छक्का है, छत पे लग के अगर हाथ में आई तो आउट है गोलू। ड्रीम लैब
Send your money from our app for fast cash pickup around the world. Send it app to cash. Western Union.
Oh, sorry. Welcome back to BioTalks. For those of you who are just joining in, we were interviewing some of the main scientists who contributed to finding different parts of the cell. Next, we shift our focus to the plant cell by talking with the man who discovered the plastid. Let us welcome Dr. Andreas Schimper.
Thank you, Mr. John. Glad to meet you. So, talking about plastids, well, it is a membrane bound organelle. Its invention was pretty interesting with my friend Ernst Haeckel. Plastids contain pigments used in photosynthesis, and the types of pigments in a plastid determine the cell color. Their origin is explained by endosymbiosis, in the act of unicellular heterotrophic protist engulfing a free living photosynthetic cyanobacteria and retaining it instead of digesting it in food vacuole. I suggest that acquisition of plastids are better described as a result of a process rather than something occurring at a discrete time and describe the shopping bag model of plastid.
That was very interesting, Mr. Schimper. That was a wonderful explanation. Next, I would like to call Dr. Albert Cloud, who discovered the endoplasmic reticulum.
Thank you, Mr. Calvin. I'm very much honored to be here on your show.
Thank you, sir. It's my pleasure too. So, sir, could you please talk about the discovery of the ER?
Yeah, why not, Calvin? Well, in attempting to isolate the Rose sarcoma virus from chicken tumors, I spun cell extracts containing the virus in centrifuges that concentrated heavier particles in the bottom of the test tube and obviously the lighter particles above, the layers of lighter particles above. So for comparison, I began centrifuging the normal cells. So this centrifugal separation of the cell components made possible a biochemical analysis of them that confirmed the separated particles consisted of distinct organelles. So such analysis enabled me to discover the endoplasmic reticulum. So there are basically two of its types, which are rough ER and smooth ER as well. And the rough ER is rough due to the presence of ribosomes on it, which enables it to the manufacture of proteins, for which are necessary for the cell.
That was a wonderful explanation, Dr. Cloud. Now, I would like to call upon Dr. Camillo Golgi to talk about his wonderful discovery, the Golgi apparatus.
Thank you, Mr. John. It was really nice to meet you at this point. Well, if you ask me how I discovered Golgi apparatus, first I should say that I studied medicine at the University of Pavia, but my study was to find if there are any cause and effect of disease or injury in the central nervous system. Many people say that they're inspired by someone, but my inspiration was a pathologist named as Giulio Bizzarro. Well, if you don't know who Giulio Bizzarro is, he was an Italian doctor and a medical researcher, and because of him, I followed researching in the nervous system. Suddenly, I discovered a staining technique called black reaction, sometimes called Golgi's method or Golgi staining. And this black reaction was a major breakthrough in the neuroscience. Then suddenly, several structures in anatomy and physiology are named using my last name, including the Golgi apparatus, the Golgi tendon organ, and the Golgi tendon reflex. Then me and my friend, a Spanish biologist named Santiago Ramón y Cajal, were jointly given the Nobel Prize in Physiology in 1906 for the recognition of our work on the structure of the nervous system.
Thank you, Dr. Golgi, for spending your valuable time with us.
You're welcome. It was really nice to meet you.
Well, this has been a roller coaster ride. So, we'll be going for another short break. This is BioTalks powered by BTech Industries, co-powered by AB Builders.
KFC. So good.
G squared, K squared, यानी घर घर की कहानी
पढ़ाई को लेकर लड़ाई वाली कहानी।
जब मैं बायजूस के मूवी लाइक वीडियोस देखता हूँ ना तो सब आसानी से समझ जाता हूँ, लेकिन आजकल के पेरेंट्स भी ना कहाँ समझते हैं।
आजकल के पेरेंट्स को भी कौन समझाए?
I thought you would say मैं हूँ ना।
अब मैं भी स्मार्ट हो गया हूँ। नाम क्या है तुम्हारा?
राहुल। नाम तो सुना ही होगा। With visualizations, concept learning and movie-like videos, Byju's is the best way to learn at home.
तो पढ़ाई की लड़ाई खत्म करो. Let's download the Byju's app now.
Welcome back and let's move on. Now I would like to welcome Dr. George Emil Palat to talk about the small but important ribosomes. Welcome to the show, Dr. George.
Good afternoon, Mr. Calvin. It sure is wonderful to be here.
So, what exactly is the ribosome?
Well, the ribosome is a complex molecule made of the ribosomal RNA molecules and proteins that form a factory for protein synthesis in cells. In 1955, I discovered ribosomes and described them as small particles in the cytoplasm that are actually associated with the ER membrane.
Okay, and what do the ribosomes do?
Well, the main function of the ribosome is for translating the encoded messages from the messenger RNA molecules to synthesize proteins from amino acids. This ribosome translates the codon, or the set of 3 nucleotides of the mRNA template, and matches it with the appropriate amino acids in the process called translation. So if this is too hard to digest, let me make it short for you. Each newly translated amino acid is then added to the growing protein chain until the ribosome completes the process of protein synthesis.
Thank you, Mr. George, for spending some time with us. Now, let's go to Dr. Anthony van Leeuwenhoek for his discovery of the vacuole. Welcome, Dr. Leeuwenhoek.
Hello, Mr. Calvin. I feel proud to stand here on the show. So, after the invention of the microscope, I examined bacteria under microscope and I was able to discover not only vacuoles but many other cellular structures too.
So, what do vacuoles do?
Well, vacuoles are membrane-bound organelles that can be found in both animals and plants. In a way, they are specialized lysosomes. That is to say that their function is really to handle waste products, and by handle I mean, take in waste products and get rid of waste products. So sometimes the waste product is water, and therefore a vacuole would have as its function to maintain the balance of water both inside and outside a cell.
Thank you, Dr. Leeuwenhoek.
Yeah, you're welcome.
Lastly, I call upon Dr. Christian Dejuve for his amazing discovery of the lysosomes or suicide bags of the cell.
Thank you, Mr. John. If I want to start talking about how I discovered lysosome, I would say that it was an accidental creation. But finding out what lysosome was really difficult because I was using the fractional technique. Me and my team were studying the role of enzyme glucose 6-phosphate in insulin synthesis. But there, me and my team failed in extracting the enzyme because of the lysosome membrane. Thus, I used centrifugation to separate cell contents. Well, the glucose 6-phosphate was detected in the microscomal fraction, the area where all the endoplasmic reticulum vessels are found. Now, this was the first clue that gave me the understanding that the enzyme is enclosed in ER vessels. However, the enzyme activity was only a tenth of the activity of a fresh sample of the enzyme. But there, I concluded that it is enclosed in a membrane that limits the accessibility of the enzyme. But after 5 days, the enzyme diffuses out of the vessels and regains its ability to catalyze reactions. And there we go, in 1955, I officially named these enzyme-containing vessels lysosome, or suicide bags. Later that year, American biologist Alex Novikov took electromicrographs of these organelles to further prove their existence. I was also awarded a share of the, Nobel Prize for Physiology for my discovery of lysosome and other organelles known as praxissomes.
Thank you, Dr. Juve. It was a pleasure meeting you.
It was a pleasure meeting you too. Thank you.
This final interview brings the show to a close. I'm sure now you have understood the main cell organelles and their functions. This is BioTalks powered by BTech Industries, co-powered by AB Builders, and this is your host John Calvin signing off. Goodbye, good night.